Explore how CRISPR converted a bacterial defense into a programmable gene editing toolkit. In this video, you'll learn the biological origin, molecular mechanism, gRNA, Cas9, PAM, real-world applications in medicine and agriculture, and the technical and ethical limits shaping its future. Chapter 1: Foundations. From DNA to bacterial immunity. At its core, CRISPR targets DNA, the biological instruction code made of 4 bases. Bacteria capture short DNA snippets from viruses and store them as clustered, regularly interspaced, short palindromic repeats, CRISPR, to recognize reinfection. The first report of these repeats appeared in 1987. The adaptive immune role was elucidated in the 2000s, and the term CRISPR was coined in 2002. In 2012, Jennifer Doudna and Emmanuel Charpentier demonstrated that CRISPR-associated protein Cas9 can be programmed with a short RNA to cut DNA at chosen sites, turning a microbial defense system into a versatile gene editing tool. Understanding this microbial origin explains its precision and limitations. Chapter 2: Mechanism. How CRISPR-Cas9 actually edits genes. CRISPR-Cas9 operates to 2 programmable components: a /20 nucleotide guide RNA, gRNA, that base pairs with the target DNA, and the Cas9 nuclease that makes a double-strand break. The commonly used Streptococcus pyogenes Cas9, SP-Cas9, requires a short PAM sequence, NGG, immediately adjacent to the target. After Cas9 cuts, cellular repair pathways determine the outcome. Non-homologous end joining, NHEJ, often produces small insertions or deletions, indels, useful for gene disruption. Homology-directed repair, HDR, can incorporate precise DNA templates but is less efficient, especially in non-dividing cells. Innovations include base editors, 2016, that change single bases without breaks, and prime editors, 2019, for versatile edits with fewer off-targets. Chapter 3: Applications. Medicine, agriculture, and diagnostics. Therapeutic and practical applications move rapidly from lab to clinic. Prominent examples include CTX-001, a CRISPR-edited treatment for sickle cell disease and beta thalassemia, developed by CRISPR Therapeutics and Vertex, which showed transfusion independence in early trial patients, 2020 to 2021. Ex vivo approaches edit patient cells outside the body, example T cells or hematopoietic stem cells, to reduce delivery challenges. In vivo editing uses AAV or lipid nanoparticles, as used in some liver-targeted trials. Beyond medicine, CRISPR enables crop improvement, disease-resistant rice, edited mushrooms, and diagnostic tools like Sherlock, Cas13, for rapid RNA detection. As of 2023, dozens of clinical trials explored CRISPR-based therapies across genetic disease and cancer. Chapter 4: Advanced topics, risks, governance, and future directions. Advanced developments raise technical and ethical questions. Technically, challenges include off-target edits, mosaicism in embryos, and efficient in vivo delivery. The adeno-associated virus, AAV, capsid limits payload to 4.7 kilobases, complicating delivery of large Cas variants. New tools mitigate risks. Base and prime editors reduce double-strand breaks. Anti-CRISPR proteins can inhibit activity. Ethically, the 2018 case of He Jiankui, who announced CRISPR-edited human embryos, highlighted the dangers of premature germline modification and prompted global calls for moratoria. Environmental interventions like CRISPR gene drives, demonstrated in mosquitoes 2015, offer malaria control but pose ecosystem risks. Responsible governance, safety testing, and equitable access remain central to CRISPR's future. CRISPR transformed a bacterial immune system into a precise gene editing platform with gRNAs, approximately 20 NT, Cas nucleases, and repair-driven outcomes. It powers therapies, crops, and diagnostics, but faces delivery, safety, and ethical challenges. Responsible innovation, rigorous testing, and global governance will determine its societal impact.